Welcome everyone to this next edition of our webinar series on optical coherence tomography. My name is Michael Leitner. I'm Product and Sales Manager for OCT at Thorlabs, and I will be moderating this live event. During the first editions of this webinar series, we already reviewed the basics of OCT and invited external speakers to talk about the fascinating research they do based on OCT. These and all other Thorlabs webinars can be found on our website at thorlabs.com/webinars or on the Thorlabs YouTube channel. Today we will discuss another very interesting application, which is the use of optical coherence tomography for biofilm research. And it is an honor for me to introduce our speaker, Dr. Robert Nerenberg from Notre Dame University in the United States. Dr. Nerenberg is a professor in civil and environmental engineering and earth sciences at the University of Notre Dame, and his research primarily centers on microbial biofilms for environmental applications. Dr. Nerenberg is a recipient of the NSF Career Award and the Paul L. Bush Award for Innovation in Water Research, and he is currently chair of the IWA Biofilm Specialist Group and an associate editor of NPJ Biofilms and Microbiomes. Please note that questions can be posted throughout the whole course of the webinar in the Q&A section, which you can access through the icon in the main ribbon. Well, with that being said, I would like to hand over the stage to you, Rob.
Great. Thank you very much, Michael, for the kind introduction and also for the opportunity to share a little bit about my research here in this webinar. I just want to point out that I would like to discuss several different applications where we used OCT. I'm not going to introduce the OCT instrument itself. I think there are other webinars that are focused on that. And I'm also not going to go into great depth in the research itself, but more focusing on how we used OCT. So with that, as an overview, I'll start out with some introductions. I'll talk about one line of research, which addresses a special kind of biofilm in a process called membrane aerated biofilms. And then I'll talk about how we can use OCT to explore biofilm deformation and couple that with models, and also a special application where we can use OCT to map the mechanical properties of biofilms. So, quick introduction. So I'm at the University of Notre Dame, not in Paris, it's in the United States. We're near Chicago here, in a town called South Bend, Indiana. And South Bend, around this time of year, actually looks like the pictures you see here. Maybe a few weeks ago, it would have looked a little colder, but it's quite pleasant at the moment. And I'm in the Department of Civil and Environmental Engineering and Earth Sciences. Notre Dame, is a medium-sized university and has an engineering college with several departments. So I guess probably don't need to focus too much on that now. Okay, so I assume many in the audience are familiar with biofilms, but still I'll give a quick introduction. So biofilms have been defined as matrix enclosed bacterial populations adherent to each other and or to surfaces and interfaces. So what does a biofilm look like? Well, let's start by looking at a suspended bacteria. So if we have single cells, the size dimension might be around 1, 2, 5 microns, and they're separate from each other. We can also have flocks. So flocks in some ways are similar to biofilms, but they're smaller and typically don't have defined structures. So they don't have as much of substrate gradients and defined microbial community structures. And finally, we have a biofilm. In a biofilm, we can have, greater thicknesses or sizes. And here we do have gradients of substrates as they diffuse and are consumed within the biofilm. The concentrations are reduced, byproducts can be produced, chemicals can be exchanged between bacteria, and usually these biofilms develop stratified microbial communities. So that's a differentiating feature. Okay, so why do we care about biofilms? Well, in the environmental engineering field, they're very important. Biofilms are often used for wastewater treatment processes. And of course, I'm very interested in membrane aerated biofilm reactors, and this is an example of one here. You can also have different types of microbial communities. On the bottom, I'm showing some algal biofilms. You can also find biofilms growing in water systems. So we know that we treat drinking water and we disinfect it, but unfortunately, biofilms can still show up in our pipes, and this can cause health problems. For example, opportunistic pathogens can develop, especially in the biofilms, and cause exposure and illnesses to susceptible populations. And of course, biofilms are also bad actors in human health situations. Biofilms can cause infections, and they're very problematic. Biofilms are very difficult to eradicate using antibiotics. They're much more resistant to antibiotics than suspended cells. And this is an example of an infection in the bronchioles. For example, for people with cystic fibrosis, they're very prone to these types of infections, and they're very difficult to remove. And here's just an image of a biofilm of Pseudomonas aeruginosa, which is typical of these cystic fibrosis infections. All right, so biofilm imaging. So we often want to see what we're working with. So we want to see our biofilm, and there are a number of ways to do that. So let me do a quick review. And I'm going to do a little bit of drawing here with my mouse, so I apologize if it's not very clear. But here on the top, we have a biofilm growing on a surface. And I'd like to show you how we can view that biofilm at different scales. So we can get a large view of the whole biofilm just by using photography. So for example, we can use or microscopy. This is with a stereo microscope. We take a picture, and here we can see the biofilm. In this case, it's growing on a hollow fiber membrane, and that's great. We can see it at a large scale. We can capture several millimeters or even centimeters of our biofilm, but we can't really see what's going on inside the biofilm. We can't see, if there's pores. It's hard to get a three-dimensional rendering with this technique. So it's limited. It's very easy. It's non-destructive, but it's a little bit limited in the resolution and what we can capture. So we can go a little bit deeper. Here, I'm showing a blow-up of what we can capture for a small section of biofilm, and that's shown here. And now at this scale, we can see much more of the biofilm structure. We can see individual cells, and we can see pores. And here's an image from confocal microscopy. And here, we can actually see individual cells. We can use staining, for example, fish or live dead stain, and we can see what the cells are doing. So we can see if they're live or they're dead, or if there are specific phylogeny. So again, this is a very detailed information, and it's great, except it's destructive. So usually we have to take the biofilm away from our growth environment. We have to fix it and stain it, basically killing the bacteria. And also the scales that we get are usually smaller, so maybe 100, 200 microns on the side. So it's a great, very powerful tool, but it's destructive. It also requires pretty expensive equipment. So again, it's good for its purpose, but it also has some limitations. We can keep going further on, and if we look here at a small part of this biofilm, we can use scanning electron microscopy or transmission electron microscopy and get even more detailed information. We can see individual cells or filaments. But again, this is even more destructive. We have to dehydrate the sample and sputter coat it and take it to an expensive, really large piece of equipment. So usually for biofilms, this involves destroying the structure of the biofilm, so we lose that original structure that the biofilm has. So maybe something in between that could capture this scale, somewhat like the light image, but with more detail, and maybe also getting interior information, is OCT. So here, we're basically imaging this biofilm at a scale of several millimeters. You can see here, the scale is about 1 millimeter. We can see the biofilm contour. We can also see what's happening inside the biofilm. There's some pore structures. We could also, and I'll show this in a second, get three-dimensional renderings of the biofilm similar to the confocal microscope. And we can actually get movies. And I'd like to mention that I'm going to be showing a number of movies in this presentation, and they'll show up in a little separate window, not on the actual slide. And I should mention also that each video will be played twice, so you'll have a chance to see it if you missed something the first time. Okay, and one final thing is it's non-destructive. So OCT can be used without taking your sample necessarily from its growth environment. Again, if you grow it in a flow cell that allows imaging, then you can image it non-destructively. You can image it over time. And it's a very simple device, very simple. It would look, I'll show it in a second. It looks simpler than a regular light microscope like you'd see in a high school. Okay, so here's my story with OCT, how I got started with OCT. So it turns out in Germany, a group of researchers at the Karlsruhe Institute of Technology, Harald Horn and Michael Wagner, I believe were one of the first people to start using this technique for biofilm research, at least in the environmental field. And they were, I believe, colleagues or good friends with researchers at EWAG in Switzerland, Eberhard Morgenroth and Nico Derlin. And I believe Eberhard and Nico, seeing how interesting this technique was, they got their own system and began using it for their research. So it turns out I was visiting EWAG and visiting Eberhard and Nico and learned of what they were doing, and I found it very interesting. So let me explain what they were doing and how I got involved in that. So they were very interested in filtration, water filtration using membrane systems. And what they found was that, you know, you usually expect filtration membranes to foul over time. A biofouling layer develops. They were filtering river water with a fair amount of organic matter. So you would definitely expect a biofouling layer to develop, and sure enough, it did. So here you can see a filter. I think this was fairly large, maybe 30 or 40 millimeters in diameter, and a biofilm developed. But you can see the biofilm is quite rough and patchy. Now, when they developed or studied a similar experiment, but this time using cyclohexamide, which inhibits protozoa, or eukaryotes in general, they found a much smoother biofilm. So they were saying, well, okay, how can we quantify the difference between this smooth biofilm and the rough biofilm? Let's use OCT. So sure enough, they did. And here's what they found. They found that, as expected, the biofilm growth with the protozoan inhibitor was much more smooth and uniform in thickness, and the one with the protozoa was much rougher. So they were interested in trying to relate this morphology to the filtration flux, so how the permeability of the system changed over time. So that's where, I guess, the moment where I came to visit and saw they were working on this and thought, well, why don't we take the images from OCT, import them into a mathematical model? And that's what we did. We took the OCT image and basically converted it to a black and white image, got the contour, and imported that into a COMSOL model of fluid flow through permeable media, and was able to find, or were able to find, that the thickness really didn't make a difference once it was beyond a certain value. So if the thickness was above a certain value, it didn't matter how thick it was, the flow or the flux was zero. In contrast, in these thin areas, there were hotspots of flow. So these thin areas had disproportionately large fluxes. So these thick areas contributed to average thickness, but didn't make a difference as far as flux. So looking at the average thickness of these biofouling layers was misleading. So again, we used modeling, and with the average thickness, you get a uniform flux, but with variable thickness represented by this kind of zigzag sawtooth pattern, you get hotspots here in the thin parts that are disproportionately high. And those govern the flux of the membrane. So again, this is how we got involved with OCT, and we were really excited. We thought this was a really neat application that allowed us to take a very large scale biofilm and convert it into information that was tractable, and we were able to model it. So again, we used many of these profiles for this research. So here's what an OCT looks like. And like I said, if you've seen a confocal microscope or an SEM, this is like a toy in comparison. So this is basically the imaging unit. It's very small, it's light, it's maneuverable, and you can bring it to your flow cell, or you can bring your flow cell here. It's quite easy to manipulate. And the cost is also much lower than a confocal microscope or an SEM. And here's just an example of some images that we got. So again, after coming back from our collaboration, we said, wow, can we try one out? And Thorlabs kindly lent us a demo, and we were able to get images like this. This is a biofilm growing on a support or a membrane. Inside that, we have finer membranes that you can see here as little circles. And here's a kind of a filamentous biofilm growing on top. So three-dimensional rendering, non-destructive, non-invasive, no staining, and very quick. And here's the scale, 1 millimeter. So we were super happy with this, and we're able to get funding and purchased our own OCT. Okay, so here's how we used it. So one area of research that we work a lot with in my lab is membrane aerated biofilm reactors, or in general, membrane supported biofilms. So let me tell you a little bit about some research in this area. So first of all, what is this membrane aerated biofilm? Well, it's a type of counter-diffusional biofilm. Let me explain. So if this is imagine this is a biofilm, this is the attachment surface. So this is like a solid surface. Here's the biofilm, and this is the liquid diffusion layer, and here out is the bulk liquid. So substrates, let's say an electron donor and electron acceptor that the bacteria grow on, diffuse from the bulk across the liquid diffusion layer and are consumed as they diffuse into the biofilm. So this is the donor, and the acceptor similarly diffuses from the bulk across the liquid diffusion layer into the biofilm. Since the acceptor and the donor diffuse from the same side, we can call this a co-diffusional biofilm. Now, what if we have the situation where we have one substrate from the bulk, in this case the acceptor, and the other substrate coming from the base of the biofilm, in this case the donor? Well, now one is coming from one side and one from the other. We can call this a counter-diffusional biofilm. And even though it seems like a very minor difference, it actually leads to very different behavior. And I'll touch on a few examples in my following slides. I just want to point out here that in a co-diffusional biofilm, the highest concentrations of substrate are at the outside. The bacteria are most active where they have concentrations of donor and acceptor that are both high, and that always occurs at the exterior of the biofilm. With a counter-diffusional biofilm, as the biofilm gets thicker, the area that has high concentrations of both donor and acceptor can occur anywhere in the biofilm, depending on the relative concentrations of donor and acceptor. But in this case, it happens near the middle, and it's actually quite low. So because both donor and acceptor have to travel through the biofilm to reach this point where they're both present, the concentrations have become lower, whereas here, the concentrations are always highest at the outside. Okay, I'll touch on that back in a second. So, where do we see these counter-diffusional biofilms? I would argue in many places in the natural environment and also in engineered systems. Here's an example of a biofilm growing on a liquid surface. It's usually called a pellicle. And in this case, nutrients come from the liquid phase, let's say organic matter, and oxygen comes from the air. And the biofilms grow on the surface because that's where they get their highest concentrations of oxygen and organic matter. And if you're a fan of kombucha, the fermented tea drink, you may know that it's made by fermenting tea with sugar using what they call a SCOBY, which is a syntrophic community of bacteria and yeast, and it grows as this very dense and very thick biofilm on the surface of this tea mixture. So again, this is an interesting case of a counter-diffusional biofilm. Now, when we grow biofilms on gas-supplying membranes, for example, if we're supplying oxygen or hydrogen or other gases, they also can be counter-diffusional. So the gas diffuses from the membrane, this is the membrane here, out towards the bulk liquid. So this is the bulk. And if there's nutrients in the bulk diffusing towards the biofilm naturally forms on the surface of the membrane. And here you can see it on this light microscopy image on the right. The membrane's in the middle, and here's the biofilm growing on the membrane. So again, these are counter-diffusional biofilms, and the bacteria like to grow on the membrane because that's where they get their highest concentration of acceptor and donor. Okay, so let's talk about some research that we did with these membrane aerated biofilms. So one topic addresses predation, so protozoan predation. This is a setup that we often have used to study biofilms, in particular these membrane aerated biofilms. It's basically a single membrane inside a glass flow cell. The membrane is pressurized with gas, and we have ports in this membrane, in this flow cell that allows us to use microsensors. So if we look at this here, we can see that this port can be opened and we can insert a microsensor and measure gradients of oxygen or other ammonium, nitrite, nitrate in the biofilm using a micromanipulator and a microsensor system. But we also can use OCT to image the biofilm in real time. So this is a system that we use for many of our experiments. And here's a picture. Here you can see the glass flow cell. Here are the ports where we can put in the microsensor. Here's the biofilm, for example. And actually, this is a microsensor that we're using to create profiles in the biofilm. And here's a picture of a pretty thick biofilm. They're not always this thick, but just an example. You can see this is a square section flow cell, which makes it nice for the OCT imaging. Okay, so here's an example. Let me show you what I'm going to, I guess I'm going to show you a movie, and let me explain it real quick before. What we're seeing here is a longitudinal cross-section of a membrane. This is the lumen of the membrane, this is the wall, and this is the other wall. So it's like we're sectioning it along its length. The biofilm is growing on the exterior surface, and here's the liquid out in the bulk. So what I'm going to show you now, pay attention here to the biofilm, and you'll see how it grows over time. These are time-lapse images, I believe every 6 hours or so. Okay, it's going to play twice, now it's repeating. Okay, great. So you can see that the biofilm grew, but I don't know if you noticed, there were actually voids at the bottom of the biofilm. So this is a blow-up of that same membrane. So here's, again, the membrane. This is the lumen. And here's the biofilm. And I want to point out that this is a void. And I'd like to show you a video of what's happening in that void. Again, it'll repeat twice. I'd like to point your attention to the interior of the biofilm and to some movement. This is a real-time movie. This is, I guess, what's actually happening. A second in the movie is a second in real time. So you can see things moving around. So this is kind of a long video. I think I'm not going to go through the whole thing, but hopefully you can see at the bottom of the biofilm, in that hole, things are moving around and moving in and out of the focal plane. So I'm going to go back to this slide. And what we found were those were actually protozoa that were preying on the biofilm from the interior. So again, we found ciliates and flagellates growing in the interior. And why was that? What we believe is, normally protozoa are thought to prey on the exterior of the biofilm. But in this case, the exterior was anaerobic. There was no oxygen. Oxygen was flowing, penetrating, diffusing from the membrane into the biofilm. So the interior was the aerobic zone. So they were preying in the interior because that's where they could grow the fastest. And we found that this had implications for the biofilm development and detachment. So because of these voids, the biofilm detached more easily, as you can see here. We also found that it impacted the microbial community structure of the biofilm, but that's, I'm not going to talk about that here. So let's move on to a different topic. I'd like to talk about some unique density profiles that we found in these membrane aerated biofilms. So again, here's another movie. And in this case, it's similar to the previous one. So the biofilm is growing here on the surface of the membrane. But I'd like to call to your attention a band of high signal intensity that will appear parallel to the membrane within the biofilm. So let's take a look. And again, it should play twice. So as the biofilm develops, at some point, you see a line, a white line develop towards the top. So we were really fascinated by that band or that line. And initially we thought it was an artifact. So we thought, oh, it must be some reflection from the membrane surface. But we actually found this quite reproducibly. So here you can see, again, the band occurring in the biofilm, Here you can see a three-dimensional rendering. So this is the membrane. And here's the biofilm growing on the membrane. And here you can see the line quite clearly. See it here too. So it's actually more than a line, it's a cylinder of dense cells. And okay, we did some epifluorescence microscopy and phase contrast and found that we also saw this layer of high cell density. So this allowed us to confirm that this was in fact a layer of high cell density. It wasn't an artifact of the OCT. What we believe is happening is because of this counter-diffusion of substrates, once the biofilm becomes thick enough, the substrate concentrations where they meet become low. And because of that low concentration, the bacteria start growing more slowly. When they're a little bit starved and growing slowly, they produce less EPS and grow at a higher density. This is what we believe. And here you can see this is the activity according to a model of a counter-diffusional biofilm, and you can see it's low in the exterior, low in the interior, highest in the middle, but not necessarily that high. Here in this thin portion, you can see there's more of a hotspot because it's much thinner in that location. So again, this is a work in progress. We are wrapping up a manuscript on this, but it's a really interesting observation, and it's something that we found with the OCT with a conventional microscope. We may not have seen this. Another interesting topic is filamentous fungi in Mabr biofilms. So we found that when growing biofilms on these membranes with high donor concentrations, massive biofilms formed, massive biofilms. So this is around 1 centimeter in dimension. And here you can see this also is probably on the order of a centimeter or close to a centimeter. And we could understand why. And they were also very rubbery and different from other kinds of biofilms that we've worked with. And when looking at this with SEM, we saw these massive bundles of filaments. So these kind of tubular structures formed in the biofilm. If we look a little even closer, we find that this surface here is the gas transferring membrane, and they almost seem like they're embedded in the membrane and growing outwards and forming canopies in the biofilm. So we were really surprised to see this. Initially, we thought maybe they were cable bacteria that can transfer electrons through conductive filaments, but they didn't match the phylogeny of cable bacteria, and the morphology was different too. But using OCT, we could also see this. And again, maybe OCT wasn't so critical, but we were surprised. We could see these filaments pretty clearly with the OCT. These are the hollow fiber membranes. This is the same image I showed earlier. And if you zoom in, you can actually see the filament and the septa on these filaments. And sure enough, we've confirmed using other tools that in fact these are fungi. Just to touch on some of the techniques we're using, we have these reactors. In this case, we're using flat sheet membranes. Looks like this. And we see, again, just a few OCT images that also they seem to grow initially as a low density biofilm, and apparently with some voids at the bottom too. Let me show you a movie. This is a three-dimensional rendering, and what I'll show you is the same image, but a manipulation where we can see it from a rotation. So let's take a look. So we're just rotating that same image, and after it rotates a couple of times, it'll take a cross-section vertically and horizontally. So you can see the biofilm is a little bit less dense at the bottom and more dense at the top. Now we're looking at planes into the, I guess, computer screen, and then up and down. And moving on, here we can see that using fish, we can see that there are bacteria, but here on the left, the green are bacteria, but using a fish probe for eukaryotes, we can see the filaments more clearly with the eukaryote probe. So again, with sequencing and with fish, we're quite certain that these are actually filamentous fungi. The question is why, and that's something that we're still working on. Okay, changing topics a little bit. I'd like to talk about how we've used OCT to assess biofilm deformation. Again, I think a really powerful feature of OCT is that we can image biofilms in real time. And if we have a flowing fluid, for example, over the biofilm, we can assess fluid structure interactions. So this is an image of the biofilm, and I'd just like to show you what it looks like when we image real time the flow over the biofilm. So this is just a few seconds. The flow started, the biofilm deforms, and then when the flow stops, the biofilm recoils. See it once again, it's deforming, and after the flow stops, the biofilm recoils to its initial position or close to that. This is a setup that we used for our research. So basically we had a flow cell similar to the glass flow cell that I showed before, but here we had a sort of a stub, and we imagine it's like a hollow tube, and we put the biofilm into the tube and extruded it, so it extended out. In this case, we're actually looking at a synthetic biofilm with bacteria in alginate. So again, here's our biofilm, here's our flow, and this is what it looked like with the OCT. So again, the flow's coming here, the support is in this area, and let me just show you a quick movie of what this looks like when it deforms. Oh, I'm sorry. I think I'm showing you the wrong movie there. I actually, let me see. Let me show you a couple of things first. So this is actually the setup and the modeling we used. We modeled the biofilm using a phase field model, considering the biofilm as a viscous material. And okay, these are our results. So we were able to measure the deformation at 3 different locations using the OCT image. And we use that to compare our model prediction, where the phase field model was built using a measurement of the viscous property of this biofilm, the synthetic biofilm, which was then added to the model. And you can see that the actual deformation and the model deformation for these 3 different locations was quite good. It wasn't perfect, but it actually was quite good. And just real quick, I'd like to show you the images of the measured deformation and the model deformation. So this is the actual OCT image. Oops, I'm sorry. No, this is actually the model deformation. And this is the OCT deformation. So again, you can see in real time how this deforms under fluid flow. So using this model, we can gain other information. We can determine the flow field based on this fluid structure interaction. We can determine deformation velocities during the deformation, and we can also determine stresses inside the biofilm. And one thing that we found is this viscoelastic behavior is actually quite important. So even in short time periods, it can't be neglected. We also repeated similar experiments, but in this case using a real biofilm. So this wasn't alginate with bacteria, it was a real biofilm. Here's the biofilm structure. It was supported on a stub similar to before, and these are the results. And real quick, I'll show you some videos. Let's take a look. So here's the OCT image, real time, showing the biofilm deformation under fluid flow. So again, this is repeating itself, but you can see the biofilm deform. And here's the model simulation of the biofilm deformation. Okay, so in any case, probably the conclusions aren't so important for the purposes of this presentation, but we were able to develop a model that could capture the behavior of real biofilms, and the OCT was a really convenient and I guess practical way to compare the model to actual biofilm deformation. So I'd like to also say a few things about a special approach we use to study biofilm deformation. So I guess early on, we were really impressed with an image that we saw of a biofilm desiccate. So here's a picture of a biofilm, and in real time, under the OCT lamp itself, we found the biofilm shrink, like visibly shrunk within half an hour. So let's take a look at this shrinkage. And please notice, as it shrinks, the OCT signal intensity increases. It becomes brighter. And we believe that's because the cells are concentrating. There's less liquid between the cells, so we get a brighter signal. So we thought this would be an interesting tool to use to map the cell concentration or cell density within biofilms. So this is a technique that's been used in other fields. So for example, computed tomography is used to map bone density, or X-ray is used to map soil properties. And those properties can then be incorporated into models that use this to consider the heterogeneous distribution of mechanical properties of these materials. So we thought, well, maybe we could do the same with biofilms. I mean, we certainly saw changes in density within the biofilm interior, and other researchers had also found that there was a proportionality between cell density and OCT signal. So our idea was, okay, let's take a biofilm, like this biofilm here, and use the OCT signal, and you can see it's quite heterogeneous. Let's look at this image here. There are areas with strong signal, and there are areas with very little signal, probably because those are voids. So if we can use this to map the mechanical properties, assuming that the cell density, which we're inferring with the OCT, corresponds with a mechanical property. So we didn't measure the mechanical properties directly, but we inferred them from the OCT signal, that we can get a better prediction of the mechanical behavior. So let's take a look at just a quick image of this biofilm deformation. So again, this biofilm deformation was what we used to compare our model to. But we also used this OCT image to map the mechanical properties into our model. Okay, let's take a look here. So here, we did a mapping of the grayscale intensity of this image, and also looked at the background and assumed that the signal from the background would be similar to water, maybe a little bit higher than water in the biofilm. And then using shear rheometry, we captured the average viscous behavior of the biofilm and assigned a function to relate the signal intensity to that average. So for example, if we had an area with very little signal, we assumed that we were at a very low mechanical property compared to the average, but if we had a high signal, we had a higher property than the average. And here we can see the map. So actually mapping the value of the viscosity for the different positions within the biofilm. And to compare the biofilm OCT image to our experimental image, since there are many different points here, in some cases they come in and out of the focal plane, we used image analysis to create a skeleton and then map the deformation of different points here on this skeleton and compared that with the model prediction. And as you can see, this is the experimental data, this is the heterogeneous model, and this is with a homogeneous model. And you can see the heterogeneous model was able to better predict the deformation of the biofilm. And again, using this phase field model, we can also infer the deformation and stresses within the biofilm. Okay, so I'm close to the end of my presentations. Thanks for your patience. I know I've covered a lot of different topics. I'd just like to mention a few opportunities and challenges with OCT. I think the things I mentioned already, I think are all great opportunities. One thing that I think is a really interesting application that I think we haven't really capitalized on, but would be interesting to do, is look at real-time growth of the biofilm. So maybe not completely real-time, but maybe capturing images every 1, 2, or 3 hours and seeing how the biofilm develops under different conditions, and also trying to capture detachment. So this is a really nice tool for capturing detachment from the biofilm. And let me show you a movie of this biofilm development. So you'll see the biofilm developing on the surface of the membrane at first. Not much will be happening, but as time goes by, a much faster development will take place. So now you can see it growing, and you can actually see a consolidation taking place at the bottom of the biofilm over time. So this is a really nice tool to explore what's the effect of flow conditions, what would happen if we had a detachment event, what are the effect of substrate concentrations, oxygen concentrations, predation. Again, a very nice tool that we can use to image, and it's non-destructive. It doesn't require any staining and can be done in situ. So as far as limitations, and I see, I'm scanning the questions box with one eye as I'm presenting with the other. I think some people have already touched on a limitation, and that is signal attenuation. So it's difficult to be confident that your signal intensity is related to a cell density because of a number of factors. So here you can see that the outer biofilm has signals, but once you pass a certain depth, there's no more signal because you can't capture through a very thick biofilm using OCT. The signal attenuates. So, can we certainly say that this signal is weaker than this signal, or is it just because it's deeper or further from the focal plane? Speaking of the focal plane, here you can see quite clearly in this image, which is of a uniform synthetic biofilm. Bacteria are uniformly distributed in alginate, yet we see a high signal intensity here and a drop-off as we get away from that band. So I think we need to be careful in using the OCT signal to interpret density, especially if we're looking at a large range of thicknesses. Also, there's limits to the OCT resolution, and if you zoom in too much, you can see kind of a granularity, and it's hard to say if this granularity is because of the resolution or if in fact there is some kind of a clumping of the cells. So these are all things I think that you really need to explore carefully before you can make strong conclusions about your signal intensity. So in conclusion, I think OCT is a really powerful tool for biofilm research. It's not going to tell you who's in the biofilm. In most cases, you need some kind of a staining or a GFP tag cell and fluorescence microscopy for that. But it's very easy to use. You can use it in situ. It's non-destructive. You can do real-time imaging. And I would say it's relatively inexpensive. And it captures this mesoscale. Like if I were doing confocal microscopy and looking here or looking here, I would see two different things, and maybe I wouldn't realize why. Or maybe I wouldn't even realize that I should be exploring these two different locations, even though, they behave very differently. So with that, I'd like to acknowledge, well, first of all, I'd like to thank you for your attention. I know virtual presentations are tedious, so thank you for staying on. I'd also like to acknowledge my research group who captured all these images that I showed you today. And with that, I'd be happy to answer any questions.
Thank you very much, Rob, for these interesting insights. And we already have several questions from the audience. And the first question comes from Tufam Le from Khalifa University. Dear Professor Robert, we are a group from Department of Chemical Engineering at Khalifa University in United Arab Emirates. We observed scaling using OCT. We also observed the voids, so the space between membrane and the scaling, as you see in the case of biofilm. What do you think could be the reason for the voids in your case? Thank you.
In our case, I think it was because of the protozoan predation. So in these counter-diffusional biofilms, where oxygen is supplied from the base of the biofilm and the bulk liquid is anaerobic, protozoa typically prefer aerobic conditions. So they actually had a niche in the biofilm interior. Now, we actually think that these voids in the biofilm were initiated by amoeba, and then these swimming, grazing type of protozoa, the ciliates and the flagellates, occupied those voids and expanded them. But I think I don't think when we did experiments with pure cultures without protozoa, we didn't see these voids form. So it's interesting with scale. With scale, it might be more challenging to see into the scale layer. If it's a very dense layer and if it's optically opaque, the voids could be an artifact of the OCT not being able to penetrate beyond the scale layer. But I'm just guessing that. I don't know. Maybe it is something that's correct.
Thank you. The next question is from Nicoletta Torja from the National Institute for R&D of Isotropic and Molecular Technologies in Cluj-Napoca. Which is the mean of the upper interrupted line in the OCT image of the biofilm inside of the reactor?
I think you're referring to that line that we saw develop over time in the counter-diffusional biofilm, this band of high-density OCT signal. And I hope I'm answering the correct question. So again, that we don't know for sure what caused that band to form, but I guess there's two possible explanations for it. One possible explanation is initially when the biofilm is very thin, there are high substrate concentrations. So the biofilm has high oxygen because it's growing right on the membrane, and it has high organic carbon concentrations because there's no diffusive barrier in a very thin biofilm. So they grow very quickly initially. And since they're growing quickly, they can produce a lot of EPS and they form a thick biofilm very quickly. Now, once the biofilm reaches a certain thickness, substrate mass transfer limitations become important. The substrate concentrations become lower in the biofilm. And since it's counter-diffusion, you have low concentrations of donor and acceptor where the two meet. So now the bacteria are growing more slowly. They need to economize their use of resources and possibly produce less EPS and therefore result in a more dense biofilm. So that's one hypothesis. Another hypothesis is that initially they form a dendritic biofilm, and that once these gradients become defined, the only place that they can grow and have still high concentrations is laterally. They can't grow up because they're going to be limited with the oxygen. They can't grow down because they'll be limited with the organic carbon, but they can grow sideways, and maybe they're filling in those spaces between filaments. I don't think that's the case, but it could be an explanation. So I hope I answered your question. If not, feel free to write again.
The next question comes from Igor Buzalevich from Wrocław University of Science and Technology. Did you examine the bacterial biofilms of one bacterial species or several?
So we've used this technique for mixed cultures and for pure cultures. And this band, this line that we saw, also occurred for pure cultures. We used Pseudomonas aeruginosa PA1 with an antibiotic, and we still saw this same behavior with the line formation. So we're quite sure it's not an artifact of microbial community assemblages. It's because the same bacteria is behaving differently because of its environment. Again, either it's producing less EPS and growing at a higher density, or possibly it's growing laterally, but it's not because it's a different kind of organism that's growing with different behavior in that particular location. Again,
thank you. The next question comes from Jana Safarik from Orange County Water District. What is the lowest resolution? So maybe I chime in here. With Thorlabs OCT equipment, the resolution depends on the system that we decide to use for the application needs. And the best resolution that we can achieve at the moment is 3 microns axially, so in vertically in air. And this has to be divided by the refractive index. So for water, this would be in the range of 2.2 microns. And what we have to distinguish here is lateral from axial resolution, because those are separate in OCT. And laterally, we can go down to 4 microns with catalog equipment, with dedicated equipment. This could be even better. The next question is from Luca Fortunato from KAUST. Great presentation, Robert. I was wondering if you have tried to link the change in biofilm morphology in Mabr to the change in microbial community?
Thank you, Luca. That's a very interesting question. I think there's definitely potential for different microbial communities to result in different biofilm morphologies. We have not specifically looked at that, although along the lines of your question, I think the morphology also is different in counter-diffusional biofilms from co-diffusional biofilms. And the reason for that is in a co-diffusional biofilm, the highest growth is always in the exterior. There's probably a greater propensity to form dendritic biofilms. Whereas in these counter-diffusional biofilms, if the growth is in the interior, you're pushing the biofilm out from the inside. And I think that would lead to a different morphology. We haven't explored that. I think that would be a really interesting thing to look into. And I hypothesize that there are differences between the two types of growth. So thanks for the question. Sorry, yeah, we haven't looked into the different types of microbial communities.
Okay, next we have a question from an anonymous attendee. In your models for thicker biofilms, how do you distinguish between no biofilm, as water, and no light getting through that area?
Okay, that's a good question. So I think if you were to use a thick biofilm, you probably would run into that problem. You would run into the problem that your signal is attenuating, and you may get dark areas that are dark because your signal isn't penetrating. In our research, though, where we tried to relate the the OCT signal intensity to the mechanical behavior, we chose thin biofilms. So we didn't have a concern about signal attenuation within the range that we were exploring. But definitely that is a concern for thicker specimens or very dense specimens.
Okay. Another question from Igor Busalevich. Are there any differences between single species and multi-species biofilms?
Yes. I would say yes. You would have probably niches forming where different bacteria become more abundant at different locations within the biofilm, depending on the substrate limitations and the morphology. But that's very specific to the type of metabolisms that you have in your biofilm and the type of growth conditions. So I say definitely yes, there is a difference, but of course you'd have to explore that for each particular situation.
Okay, the next question is actually coming from your former co-worker Marcelo Aibar from Universidad San Sebastián. Great presentation, Robert. Do you think there is a way to couple the OCT with another technology to somehow identify bacterial species in OCT images? For example, something that generates a change in specific bacteria density?
Well, first of all, I'd like to acknowledge Marcelo because he's the one that captured those images of the protozoa swimming in the void. So he's a former PhD student and really got us excited about the OCT. So thanks, Marcelo. I think that's an interesting question, and we haven't looked into that. But I guess the question is there a way to use in some way the OCT information to identify the type of microbial growth within a biofilm? I'd say, if you were able to relate cell density to a certain type of microorganism or maybe filaments, it's possible that you could. But since we can't actually see cells, in most cases with OCT, we're not able to capture the morphology of single cells. We can't use that as a way to differentiate bacteria. But it would be interesting to see if there are other types of ways to use OCT. It's possible that you could use some kind of a fish type stain, something that would make the cells more opaque when this something hybridizes either to the cell surface or to the interior, that would allow you to visualize that with the OCT. But again, we haven't looked into that, but it'd be really nice if that were possible.
Okay, the next question comes from Zhujun Ding from Euroimmun in Germany. Hello, thanks for the presentation. Do you think that AI, so probably artificial intelligence, can be used to detect more information in the application of deformation of biofilm?
Yes, I do believe that. But I don't know exactly, I guess for artificial intelligence, you have to think of a series of experiments that would, allow artificial intelligence to understand the factors that determine the behavior of the biofilm. We haven't looked into that, but I think that's a super interesting application, and I think someone should look into that. So if you're interested in that, please let me know how it goes, or maybe we'll look into that one day. Thanks for the question.
Okay, the next one is from Sangli Srinivas from Indiana University. It's not really a question, more please combine fluorescence microscopy with OCT. So it's more
okay.
Okay, and we also have Michael Kühl from University of Copenhagen. Great talk, Rob. Could you comment on post-processing of OCT data to extract surface area, porosity, etc.? Any recommendations for software tools? Cheers, Michael.
Michael, thank you very much for the kind comments and for the question. I assume you know the answer to this question as well, but of course, there are a number of imaging processes tools that can be used for the OCT images. I think we did a lot of ImageJ, Fiji, MATLAB add-ons. I couldn't say specifically the ones that we've been using, but I'd be happy to discuss later on or refer you to some papers where we have this information. The mechanical properties one, both the phase field model and the mapping, those have both been published recently in biotechnology and bioengineering. I think the references are in some of the slides. But again, if not, I'd be happy to share them to you, and those methods are described there as well. I think some of the same tools that we use to determine, roughness, areal enlargement, porosity can be applied to the OCT as well. You have to just be careful with thresholding and things like that. But yeah, it boils down to image processing like we would use with other tools as well. Thank you.
Okay, and we have one more question from Nicoletta Toja. It's a more practical one. How do you decide that you are in focus? Do we have to use a 1D mode and follow the aspect of interferograms? Or how do you do it?
Wow, that's a good question. Maybe Michael or Sebastian, maybe you have a better answer than I can provide for that. Or if not, I can give it a shot.
Maybe your best practice first.
So we know the range where we would expect to see drop-off of the signal. And I think unless you're seeing really high gradients of signal naturally, you can see that in your image. So how do you know if the maximum signal intensity is the focal plane? I guess you don't. It's possible that your focal plane is in an area of lower intensity or lower density, and then your signal is increasing outside the focal plane, and it appears to be the focal plane when it's not. So that is true. That is a problem. I think we've avoided that mostly by not using OCT images where we have significant drop-off in the signal. But I don't know how you would determine exactly where your focal plane is just by looking at the image.
So
my
best practice advice would be actually to make use of one of the artifacts that we usually get in OCT images, which is the so-called autocorrelation. So we have several webinars on OCT technology already available. And during one or two of these webinars, we talk about autocorrelation, which actually happens if the features within the sample interfere with each other. So normally in OCT, we have both arms of the interferometer. It's the reference arm and the sample arm to interfere with each other. But if structures from within the sample work with each other, then this suddenly pops up at the very top of the OCT image. So when you go down with your focal plane, you will see that several structures will appear or disappear right at the top of your OCT image. And once that happens, you know that these features are in focus. If you're interested, please drop a line at oct@thorlabs.com and we can discuss this in more detail.
You can also scroll through the depth of the biofilm to see what the signal intensities look like when you have the focal plane at different positions. to get an idea if there is actually a drop-off or an increase in the signal intensity. And then maybe you can use that to infer where you are in your biofilm depth with the focal plane.
Great. So we already reached a full hour. So many thanks again to Rob and everybody in the audience for attending our webinar today. And if you have any ideas for OCT-related topics that you would like to see covered or discussed, please drop us a message at oct@thorlabs.com. And the next Thorlabs webinar is scheduled for April 20th, when our colleague Dr. Carl Borgentun from Sweden will introduce the new Redstone optical spectrum analyzer. I'm looking forward to seeing you again during our future events and wish you a very nice day.